Hi friends, I am Koil ma'am. Welcome to my channel Biology World. Today's topic is chromosome. Before we going to start, I will request you, if you not subscribed my channel till now, please click on subscribe button and press the bell icon to get my further videos. So friends, let's start the topic. The word chromosome is derived from two different Greek words. One is chroma, which means color, and another is soma, which means body. So the chromosome means colored body. A chromosome is a carrier of genetic material which directly involved in determination of organic development. Chromosomes are fibrous structure. They are present in each living cell. Due to the presence of nucleic acid, they can be stained by basic dye. Though the basic structure remains always the same, chromosomes differ in forms during different phases and subphases of cell division, such as in prophase, they take very poor stain due to the thread-like coiled structure, but in metaphase, chromosomes are short and condensed, and that's why they take a brightly stain and their numbers are easily detectable. So, for a better study of chromosomes, cells are arrested during metaphase by some chemicals. Collagen is a chemical very commonly used for this purpose in human and for other higher animal as a metaphase arrestor. A metaphase chromosome contains a primary constriction or centromere. See, this is the primary constriction and this is the centromere. A secondary constriction, which is also known as nuclear organizer, is also found in some chromosomes from which nucleolus formed. That is the secondary constriction and this is the nuclear organizer. In human karyotype, 13th, 14th, 15th, 21st and 22nd chromosomes contains nuclear organizer within their secondary constriction. This region carries some genes for rRNA synthesis. Except that there is a tennis ball like structure at the end of the secondary constriction known as satellite. This is the satellite. The terminal part of chromatids known as telomeres. This one is the telomere part. H. Muller first described this region. This region reveals some interesting features. Generally, two chromosomes never fuse together in the telomere portion, but when this telomere region damaged by X-rays, chromosomes form a sticky end and two different chromosomes can be fused together by this sticky end of their telomere. So now we will discuss about the physical structure of a chromosome. At mitotic metaphase, a chromosome consists of two symmetrical structures called chromatids. Both chromatids are attached to each other by centromere. See, this one is one chromatid and this one is another, and this is the centromere region. Depending upon the position of centromere, the chromatids of one side with short arm is called P and the long arm is called Q arm. This centromere region is a highly repetitive sequence of DNA. This region can deeply stained by basic dye. That's why this region is known as heterochromatin. So now what is chromatin? The nucleoprotein material of the chromosomes is referred to as chromatin. Chromatin consists of nucleic acid and protein. Nucleic acid are of two types, DNA and RNA. Protein Protein could be of two types, histones and non-histones. Now, the chemical structure of chromosome. As we know, every living cell starting from bacteria to human contain chromosomes, but they are different in physical and chemical property. Chromosomes are mainly consist of nucleic acids and protein, among which 90% of nucleic acids is DNA and 10% is RNA. Proteins are mainly histone, but some non-histone proteins are also present. Bacterial cell does not contain histone proteins, so their genetic material or chromosome is called naked. The main structural unit of chromatin is nucleosome. So now, what is nucleosome? When isolated chromatin is examined by electron microscopy, it is found to consist of a series of ellipsoidal beads. It's about 11 nanometer in diameter. This beads or chromatin subunit is called the nucleosome. You can see that here this is the chromatin or chromatin fiber, you can say. But when we study in under electron microscopy, we can see that this is the linker DNA, this is the linker DNA, and these are the nucleosome. Okay, and this is about 11 nanometer of these beads. These beads are of 11 nanometer. After partial digestion of DNA in chromatin, it reveals that approximately 200 base pair DNA is associated with each nucleosome. After extensive nuclear digestion, a 146 BP long segment of DNA remains present in each nucleosome. This nuclease resistant structure is called the nucleosome core. Okay. Now, the arrangement of histone protein in core particle is like as entry H2A, H2B, H4, H3, then repeatedly H4, H2B, H2A and exit. The H1 protein is generally present at the joining portion of two DNA in the nucleosome. It stabilizes the nucleosome. The two nucleosome particle is generally linked by a 54 nucleotide base linked DNA called as linker DNA. You can see here as linker. The length of linker DNA may be up to 144 nucleotides. That means it is varies from 54 nucleotides to up to 144 nucleotides. Now, the question is how the histone octamer forms. Okay. At first, one molecule of H2A and H2B make a dimer. You can see H2A and H2B make a dimer. Such two dimer, this one and this one, this two dimer forms a tetramer. Okay, you can see this as tetramer. One secondly, on the other hand, two copy of each H3 and H4 form another dimer. This also repeat like this way. So H3, H4 form another dimer. Such two dimer also make a fusion and make a tetramer. So here is one tetramer and two tetramer. Now the two tetramer fused each other and form the histone octamer or core histone. Now chromosomes, depending upon its characteristics, it's of two types. One is autosome and another is sex chromosome. Autosome, this type of chromosomes that contain genes for anything that doesn't relate it to sex determination. And sex chromosomes are also known as allosome or heterochromosome. These are the chromosomes which determinates the sex of that individual. But depending upon the length of the chromatid or the position of centromere, chromosomes are of two types. Firstly, you can see metacentric chromatid. In this case, both sides of centromere are equal in length. During anaphasic movement, they looks like V-shaped structure. For example, first, third, 16th number of human chromosomes are this type of chromosome. Secondly is submetacentric. In this case, this type of chromosome contains slightly unequal two arms in two sides of centromere. So, during anaphasic movement, they looks like L-shaped structure. For example, maximum human chromosomes are this type of chromosome. Thirdly, acrocentric chromosome. Here, two arms of chromosome on the two sides of the centromere differ greatly in length. One arm is comparatively much longer or shorter than the other. During anaphasic movement, these chromosomes assumed J-shaped. Generally, this type of chromosome contains the satellite portion of chromosome. For example, 13th, 14th, 15th, 22nd and 21st number of human chromosome are this type of chromosome. Now further, and the last one, telocentric. During anaphasic movement, it looks like I-shaped or rod-shaped due to the presence of centromere at the terminal end of the chromatid. This is very rare type of chromosome in animals. Thanks and wait for my further video.